All right. So today we're moving into blood. All right. And when you think of anything, when you think of blood, the primary thing you think about is it carries stuff around throughout our body, right? So that's is the primary function. It's also the medium through which we maintain the stability of interstitial fluid, because whatever is in the blood ends up going into the interstitial fluid minus a few things. And we'll cover that when we get to the vascular chapter, or the vascular module. So it helps maintain our extracellular fluid volume and also distribution of heat, right? Because if you don't have blood flow going to your hands, we did this with a couple people, Isaiah yesterday in lab. If your hands feel cold, that means not enough blood is getting there because the arteries are kind of clamped. So if you open those arteries, more heat gets to an area, right? So distribution of heat also. All right, so what is blood? If you weigh 100 pounds, it's about 8 pounds of your weight. All right? And the average adult has somewhere between 4 and 6 liters of blood, which is, if you take a Mountain Dew 2-liter and get about 2 to 3 of those, that's about how much blood you have in you. 45% of that, cells. And 95% of that 45 is red blood cells. So almost all of the cells in your blood are essentially red blood cells, which we know the function of is to carry oxygen. But we also have white blood cells, which we'll talk about today, and platelets, which take part in hemostasis. The rest of it is the liquid, and the liquid is plasma. And plasma is water and all the other stuff that's dissolved in it. All right, I'm not going to go through that list. You guys can read. But anything that you can dissolve in water is dissolved in the water and is considered plasma. Right? So that's waste, nutrients, things like that. This is a good summary slide of everything that's in your blood. All right, so again, one of those slides where I forget what is in the categories are. Good snapshot. So we're going to cover each one of these areas in a little bit of detail as we go along. Starting with plasma. All right. So, again, water, the stuff specifically that's dissolved in there, and we're going to go through some of these. Plasma proteins, we're going to go through a couple different types, what they do, why they're important. Gases and nutrients, we already talked about that when we did the respiratory system, right? What two gases are primarily concerned about? O2 and CO2, right? But you also have nitrogen dissolved in there because most of our atmospheric air is what? Nitrogen. Waste substances. Amino acids aren't waste, those are nutrients, but the rest of the stuff on that list are waste. As well as electrolytes, and we're not going to get into electrolytes here. Do we get into electrolytes at all? I don't remember. But electrolytes, sodium, potassium, you guys have probably had this before. Hyponatremia, hypernatremia, hypocalcemia, hypercalcemia. And you want to poke your eyes out with a spoon. But it is important stuff medically, so you do have to know that. Serum. You've heard of serum. Serum is plasma, essentially, without the clotting proteins. So if you take a tub of plasma, let it coagulate, take out the solid, it won't be real, take out the mushy, gross part, the liquid left over will be serum. All right, so what are the plasma proteins and what do they do? Your primary plasma protein is albumin, all right? And that's going to be a very important protein medically, too, because you want to know your albumin levels because that helps you determine acid-base disorders as well. We're not going to get into that calculation, but on your labs that you have, you will see albumin levels. All right. Their function, they maintain osmotic pressure, so in order to keep the fluid, or in order to keep the water inside the blood vessels, you need albumin to pull that water in, so it kind of keeps it in there. And it transports molecules, right? So we know that cholesterol is fat-soluble. It needs to be transported through the blood. We know that bile is fat-soluble, needs to be transported through the blood. Other fat-soluble things need to be transported through the blood, and albumin is one of the proteins that does that, all right? Really important, if you, it's produced by the liver. So if you have liver dysfunction, what's going to happen to your ability to keep water in the blood vessels? Are you going to be able to keep water in the blood vessels? No, so where's it going to go? Pardon? To the tissues, interstitial fluid, right? What's that going to cause? Swelling, right? Edema. Good. Fibrinogen is another. I'm sorry. It's covering letters. I'm really anal about that kind of stuff. So I'll fix that. Fibrinogen and other clotting proteins are also proteins that are in the blood always. They're always in the blood. We'll talk more about those later. Most of those, there are only a couple that are not, are produced by the liver. Again, if you have liver problems, you're going to have clotting problems too. All right, then we have our globulins. All right, the most famous globulin that you probably all know about, and you've had a whole section about it, and there's different types, and they do different things related to their main function, are your gamma globulins. Those are your antibodies, right? Those are always going to be, they're always fighting. Question for you. If antibodies help protect against disease, what specifically do antibodies do? Pardon?
Are they a marker? Like they say this is what needs to be destroyed?
Right. They bind. Nothing more. They bind. They don't eat things. They don't destroy things. They bind and send a red flag going, hey, this needs to be destroyed. You can take me too. So we'll talk about that when we get to the immune system in more detail. Not produced by liver, right? Those are produced by liver tissues. Alpha and beta globulins, again, produced by liver. So most of your plasma proteins are produced where? Liver. Liver dysfunction means plasma protein dysfunction. All right, so these alpha beta globulins, those act like albumin and transport fat-soluble things. They're not as abundant as albumin, so they're not going to have as much of an effect on osmotic pressure, but they will transport fat-soluble substances. All right, we're going to see this again at some point, but this is what body fluid regulation looks like, and kind of what I was trying to get at when we said if you don't have plasma proteins, where does the water go? So we take water in, fluid, right? It gets absorbed by our digestive tract, which is why we did the digestive tract first. Goes into the blood as plasma. That water then gets into interstitial fluid, and from the interstitial fluid, that gets into cells. Okay? Likewise, if we have too much water, it goes back and we get rid of it. Right? To the plasma, then we get rid of it in the kidneys, and the kidneys give it to the bladder, and then the bladder pees it out. If we start losing excess water, right? We start losing. We're not taking any in. We're losing water. We're going to lose it from plasma first, right? So we sweat. The sweat gets its fluid from the blood, and so we're going to lose it from the blood. Well, we need to maintain that blood volume, so what's your body going to do? It's going to pull water from your interstitial fluid. Well, then you need to fill back up your interstitial fluid. Where's water going to come from? Intracellular fluid. And that will keep happening until your cells go shrivel up, which is death. So it's important to realize that if you lose water or gain water in one compartment, it's going to affect all the other compartments. All right? Really important, and that's going to be really important when you're giving IVs and what type of IV fluid you're giving. We're not going to talk about that, but I want you to be familiar with that concept. All right, let's talk about red blood cells. RBCs, erythrocytes, all mean the same thing. All right, your red blood cell. We know the structure. They are one of the smallest blood cells, biconcave disc, so like a little donut with like a membrane for the hole. I only put the dimensions here. One, to back up an argument I make later, and two, because sometimes on your lab values you will see the dimensions, right? And the dimensions will change when you have like anemias and things like that. So I'm not going to go into it now, but that's why it's there. All right, it contains hemoglobin, right? And hemoglobin is the molecule that allows us to bind oxygen. And I hate the way I've looked. I spent hours, actually, not this time, but throughout my career teaching this, looking for pictures that show an accurate depiction of what hemoglobin looks like in a red blood cell. Can't find it. I'm going to have to draw it or something. But they draw it as if there's one hemoglobin molecule in a red blood cell, right? But hemoglobin is a tiny molecule relative to the blood cell. So there's thousands, millions of hemoglobin molecules inside red blood cells. So much that it takes up about 1/3 of the total cell volume. So when we talk about 4 oxygens binding hemoglobin and then they release, we're talking about all the hemoglobin. That happens across all the hemoglobin in a red blood cell, across all red blood cells. Okay? So keep that in mind. It's not just one hemoglobin molecule. And I realize I may sound like, duh, we know this. But I want to put it in perspective. All right, red blood cells, also one of the only cells in our body. I think there's one other, but I can't remember what it is, that doesn't have a nucleus. So if you don't have a nucleus, can you divide? No. So you have to constantly be producing red blood cells. They don't have mitochondria, so that can they do a whole lot? No. They don't have the ability to make energy. All right? So what that means is that if they can't divide and they can't produce energy, they're going to have a really limited lifespan. Anybody know what the lifespan of a blood cell is without looking ahead? Pardon? 120 days, which is about 30, 60, 90, 100, 4 months. Does that mean that you have 4 months worth of red blood cells produced, they die, and then you produce 4 months worth? No. Okay? It's a constant turnover. You guys are looking at me like, no? I was like, that's kind of stupid. All right. This biconcave shape, it is made because of the cytoskeleton, the way the cytoskeleton is arranged within the red blood cell as the red blood cell is being produced. But why? Why do you need this donut? Why do you want a donut circulating around? Well, the primary importance for that is that it allows red blood cells to kind of fold over really easily and squeeze through really tiny capillaries. All right, so that's the primary function of why it is that biconcave shape. But as a consequence, that also places that third of the volume about 30. Do you guys know what a cubic millimeter is in actual volume units? What's a cubic centimeter? Like, I'm going to give 1 cc, a milliliter. So a cubic millimeter would be, go 3 units down. It's Friday. It's Friday. There's a radio show out of Detroit, and they always go, "It's Friday, bitches." Anybody know what I'm talking about? No. Because you listen to country. Microliters. So a cubic millimeter would be microliters. Cubic centimeter would be milliliters. Lost track. So that 30% of the volume of the red blood cell in a biconcave shape will place that hemoglobin really close to the surface of the membrane. That will lower the distance for diffusion and make it really efficient to transport oxygen. All right, normal red blood cell counts. You don't have to memorize them for my class. I honestly don't care. But you should recognize, but at some point you will have to know them. And as soon as you know them, you're going to move to a new hospital system and those values change. Right? It's going to be different everywhere, slightly different everywhere you go. I'm like, son of a... But the good thing is, you really don't have to remember them. Like, somebody in this program will tell you you have to remember them. I'm telling you you don't have to remember them. You know why? Because they'll always appear on your chart. They'll always give them to you. Men have more than women. And children have slightly less than women. And I forgot to put the elderly values in here. But elderly is also slightly different as well. All right? So what is hemoglobin? Concentration 34 grams per 100 milliliters of packed cells. So if you take all the hemoglobin out of cells, 100 milliliters of packed cells, and weigh it, it'll weigh 34 grams. All right? What that adds up to, you guys, we'll go over it in a second, but do you know what a hematocrit is? You know of it. All right? If we take our total blood volume, like put blood in a tube, let's say it's 100 milliliters, and put blood in there, spin it down, and this bottom part is red blood cells. RBC volume. RBC volume over total volume is your hematocrit. So it's percent red blood cells to total blood volume. All right. Normal's about 40 to 45%. That also varies a lot between individuals, between men and women. It's slightly lower in women because we know women don't have as many red blood cells, generally speaking. Okay, the rest of these, I don't care that you know the values, so I'm just not going to cover it. It's there for reference. All right, here's your hematocrit. I already explained what a hematocrit is. One thing that they don't talk about in a hematocrit that's not really important, but it's there, is that, remember, 95% of your cells in your blood are red blood cells. The other 5% are all the other white blood cells, every other white blood cell. Like that little blue line, that's all your white blood cells, okay, compared to the red blood cells. We call that the Buffy coat, and that's always going to be at the top. All right, so we went over hematocrit, what that is, what does it mean? All right, so these are 3 tubes. On the left, we have normal, all right? So that should be about 45%. All right. In the case of anemia, what happened? You have less red blood cells. Let's call that maybe 25%. Okay, I'm making shit up. Okay, just making numbers up. But, well, making this number up, not this number. 25%. So what we call that is a low hematocrit. You have less red blood cells compared to the total volume. Notice that the total volume is still the same, right? Less red blood cells per volume. All right. We also can have conditions where we have more red blood cells per unit volume. One condition of that is polycythemia. Anybody know what that is? Break down the word. Poly, many, cyte, cell, emia. Many cells in the blood, right? Polycythemia. What kind of cells? Red blood cells. Sweet. And you guys said that med term quiz exam was hard.
You should take it. Some of it wasn't. Some of it wasn't really midterm.
We don't talk about Bruno. No. All right. So here's a question for you. What do you think your hematocrit would look? So, I mean, and these are documented. You could find these. Do a hematocrit. You suspect these things, or to diagnose these things. If you get a hematocrit, how would you know somebody is dehydrated? What would that hematocrit look like? It would be high, not poly. Polycythemia is a very specific thing. But it would be high. It would look like this. It would look like polycythemia because although your red blood cell volume hasn't changed, the amount of plasma you have has, right? So you have less volume per less liquid volume, more red blood cells. So they're more concentrated is what that's saying. Okay? So if you're overhydrated, low, right? It's all a dilution thing. All right, so where do red blood cells come from? Right? We said they're constantly produced because they can't reproduce themselves, so they have to come from somewhere. They come from these stem cells called hematopoietic stem cells. Say it with me. Hematopoietic. People fuck that up all the time. Hematopoietic stem cells. From red bone marrow. All right, from red bone marrow. Why do we say red bone marrow? Because there's also yellow bone marrow. What's that made out of? Fat. All right, these hematopoietic stem cells have a bunch of receptors on them that respond to different hormones and growth factors. All right, and they can potentially become any of the cells that make it into the blood. Okay, so that's what these hematopoietic stem cells do. They go down a number of different differentiation pathways. It's a tongue twister. What am I thinking of? Platelets, red blood cells, white blood cells, platelets, all the different types of white blood cells, things like that. All right. Within the first few weeks of gestation, like, you know, after fertilization, you guys just had that shitty lecture. They're found in the yolk sac, and we didn't discuss yolk sac. We're not getting into embryology. Like, we don't have time for that. It's neat, but also found in the liver, spleen, and lymph nodes about mid-trimester, right? So developmental pathway. But in the adult, from like just before you're born until the day you die, you'll find it in red bone marrow. All right. So that's what we're mostly going to be concerned about. So where is that red bone marrow? Where do we get it from? Well, if you're young, like 25 to 30 or younger, We can find it in the tibia and the femur, in the shafts, in the diaphysis. Okay? But after about 20 to 30 years, the shaft of the femur and tibia starts to turn to yellow bone marrow. Like it becomes more fatty. So then we rely on red bone marrow to come from ribs, sternum, vertebrae. What do we know about those bones? I think you covered this in the first 2 weeks. What kind of bones are they? Are they long bones? They're not, well, some of them are, they're irregular or they're flat, right? So a lot of trabecular bone, a lot of that spongy bone with holes in it. This is why I love to get to know you guys. Fuck you, Dr. Ways. All right, so this is kind of the better version of what I tried to draw here. Here's your hematopoietic stem cell. Here's the red blood cell pathway. Here's the platelet pathway. Here's the white blood cell pathway between your granulocytes and your agranulocytes. Okay? I wrote in, if you want the stem cell to go down the red blood cell pathway, the hormone erythropoietin, EPO, binds to a receptor on that cell, says, hey, let's activate the genes that will put you down this pathway, and you'll make a set of sub-stem cells that will head down this pathway. Similarly, you have a different receptor for thrombopoietin. Thrombopoietin will activate genes, create a sub-stem cell, will send it down the megakaryocyte platelet pathway. Okay? So it's all dependent upon what your body needs, what your body's telling these stem cells you need. All right? Again, these stem cells are in red bone marrow. All right, so since we're talking about red blood cells, let's stick with red blood cells. I don't care that you know the different stages, thank you. The different stages of that differentiation pathway. But I do want you to know that about at the reticulocyte level, because you'll hear reticulocytes, those are immature red blood cells. That's about when they say, get rid of their nucleus and start to mature into that biconcave shape. All right. If this differentiation doesn't happen properly, or you have defective genes going into making those red blood cells, then you get different types of anemias. All right. Some are, these are terms that you'll have to know eventually. Microcytic, hypochromic. What does that mean? Microcytic means? Microcytic, small cell. Hypochromic. Compared to this? No. Color. Low color. Small cells with low color, right, is one possible. That's a type of anemia, microcytic hypochromic anemia. And specific anemias have that characteristic. Sickle cell anemia. We'll talk a little bit about that. That's a I thought one of you guys were talking. I was like, who has the question?
Sickle cell.
Yeah, sickle cell. I can't talk and chew gum at the same time. Sickle cell anemia, defective globin gene, which will cause, under certain conditions, the hemoglobin to alter its conformation, which will change the shape of the cell and make this sickle appearance. All right? And the problem with that is it's not effective at carrying oxygen, but it also gets stuck in blood in small capillaries. And if you get stuck in small capillaries and nothing can get beyond that, you just deprive the tissue of oxygen.
Is that what causes the pain?
Yep.
The same thing later things. The ischemia is what causes
Right.
it.
Yep. Megaloblastic anemia. Big cells. Megaloblastic anemia. And erythroblastosis fetalis. I don't expect you to know what that means. That's a consequence of if a mother's immune system attacks a baby's red blood cells because they're Rh incompatible. You know about that, right? We'll talk about that later if you don't. It'll produce that disease, leading to an anemia. So different types of anemias related to formation of red blood cells. But this isn't pathophysiology. We're not going into too much of that. All right. How do we get the production pathway started? Our body has to detect, okay, what do red blood cells do? They carry oxygen. All right, they carry oxygen. So, when would you need more red blood cells? When you need more oxygen. When your body is low in oxygen. So when your blood is low in O2, there are certain cells in the kidney that detect that. Kidney. There are some in the liver too, but by far it's in the kidney. In fact, when almost 20 years ago, I was teaching this, and I said, all right, it comes from the kidney and the liver. And he goes, since it comes from that, can we call it the cliver? Okay. So that became a running joke in that class. So when the kidneys detect low blood oxygen, the kidneys will release erythropoietin. So one of the things that I want you to get in, stuck in your head is that not all hormones, and we saw this in the last chapter too, 2 chapters ago, not all hormones are secreted by endocrine glands, right? Kidneys can secrete hormones. So they secrete erythropoietin. That goes to the red bone marrow, to those hematopoietic stem cells, starts making more red blood cells. You get more oxygen in your blood, which will eventually go by the kidney again. The kidney says, hey, we have enough oxygen. Let's shut off erythropoietin, and you don't make any more red blood cells. Okay? What kind of pathway do we call that? Where it will stimulate the release of something and shut itself down. Negative feedback. Good. All right. So what are some factors that will decrease oxygenation? Low blood volume, right? If you bleed out, you don't have as much blood, you don't have as much red blood cells. You're going to stimulate the kidneys to make more. Anemias, low hemoglobin, no matter what the factor, low iron, low ability to make hemoglobin, things like that. Poor blood flow to the kidneys specifically. If the kidneys aren't detecting oxygen, then they're going to make more red blood cells, even though your body has enough red blood cells, right? So that can become a problem. That's when you might get something like polycythemia. And pulmonary disease, and I don't think I need to explain that because that makes sense, right? If you have problems breathing, you're not going to have oxygen. So, within minutes to hours of detecting low oxygen, erythropoietin will get secreted. All right, within minutes to hours. And keep in mind, like, we're constantly killing off red blood cells. They're constantly dying in droves, right? So our kidneys are constantly detecting the fact that we have lower oxygen, so increasing the production. So don't think that this is something that just happens in response to a pathological condition. This is constantly going on. All right, so we get new circulating red blood cells in about 5 days. All right, so we detect it in a couple minutes to a couple hours, but it takes a couple days for those red blood cells to get out. All right, so there's a time lag. All right, that's important to consider when we're talking about blood donation. That's why they say you have to wait so many days after you donate blood to donate again, because they take at least 5 days to repopulate.
Is there something else that takes longer? Because don't they usually tell you like you have to wait like close to 3 months almost?
I don't know what the wait time is. Is it 3 months?
3 months.
That has more to do with the fact that the amount of red blood cells that you just took out of your body, more so than this normal turnover. Okay, and if you do blood, okay, so you go give blood. This would be a cool experiment. I mean, it's already been done, but I'd still like to do it. Go take some of my blood, test it, look for erythropoietin levels. A couple hours after you took my blood, take more blood, a little amount, test for erythropoietin levels. And then 1 week, 2 weeks, 4 weeks later, check for erythropoietin levels. And you see, like here, then what would it do? Up, then maybe up a little more, then start slowly coming back down as oxygen levels get replenished. Eventually till it gets back to where it should be. Should be basal levels. All right, so what's important for making red blood cells? Number one, vitamin B12. We talked about this already. What's important for absorption of vitamin B12? Intrinsic factor. Where's intrinsic factor made? Stomach. Pardon? In the parietal cells. Good. In the stomach. Where is it absorbed? Ileum. Good. Not small intestine. You guys know better than that. The distal ileum. Okay, so very important. And the importance of it, B12, is required for DNA synthesis. Why is DNA synthesis important? Because you need to divide to make more progenitor red blood cells. All right, so the better able you are to divide, the more red blood cells you can produce. Folic acid, similar thing. Iron, obviously that's necessary because that's necessary to make hemoglobin. And vitamin C. And that helps with the absorption of iron. So even though all of these are needed for red blood cell synthesis, some are directly involved, some are indirectly involved, like vitamin C. All right, so hemoglobin is formed from early stages all the way up through where the reticulocyte is. So you constantly form a hemoglobin through there. Yeah, that's all I want to say about that. So, what is it? You don't need to know all this. Huh? Cool. You've heard of succinyl-CoA, though, before, right? Where's that found? Where's the Krebs cycle happen? Mitochondria. So, where is hemoglobin made? Mitochondria. Yep. Sweet. All right. So, basically, we have mitochondria putting things together and making these hemoglobin molecules. This is what a hemoglobin molecule looks like. Okay? Iron is at the center right here. Okay? And then this hemoglobin molecule will get put into a single polypeptide chain. And another hemoglobin molecule will be put in another polypeptide chain, another one in another, and another one in another. So each hemoglobin molecule has 4 polypeptide chains. Each polypeptide chain has 1 hemoglobin, and each hemoglobin has 1 iron. So how many total irons? 4, right? 1, 2, 3, 4. So that means you can bind a total of 1, 2, 3, 4 oxygens. What's the charge on iron? Positive. What's the net charge on oxygen? Negative. So they're attracted by electrostatic forces.
They're asking, they're talking about something about pickup truck and
Oh. I didn't park next to a pickup truck. Okay. I've got a minivan. Awesome. I raged against that at first, but then we got the ones with all the bells and whistles, and I called it the Starship Enterprise. My only Star Trek reference in my life. And now it's cool. Now it's real cool because I have I had kayak racks. And now I throw them up there, throw stuff in the back. It's great. Like, I love a minivan now. What I won't do is pay the amount of money they're asking. I would love a truck. I will not pay the amount of money they're asking.
It's insane.
F you. No. You don't get my money if you're going to rate me about the price. All right. So these globin chains, they're polypeptides, right? They're proteins. You have genes that make mRNA that make the proteins. So these polypeptides come from proteins. And we have two types that are normally in our hemoglobin, which is hemoglobin A, alpha and beta chains. All right, 2 alpha, 2 beta. Okay, that's why you have this color difference here. Each one still binds a hemoglobin the same. Each one still binds iron the same, and will still bind oxygen the same. Okay? Where we run into problems is when we start having, not problems, when we have different chains. The different chains that can get incorporated, and they get incorporated in different genetic backgrounds, different stages of life. But the differences they have in O2 carrying capacity is relatively minor, right? So if you don't have alpha, beta, you have like delta, you're not going to have much of a difference in carrying capacity. But if you have mutations in some of these that even change one amino acid at one position, that can change enough to cause a disease that will affect the rest of your life, which to me is mind-blowing. And it's not even one amino acid. It is one base that's changed to change that one amino acid. And you've got this screwed up disease the rest of your life. So that sucks. So that's sickle cell anemia. Normal red blood cell, sickle cell. You'll talk about sickle cell crisis at some point. So these sickle cells, when they start going down smaller capillaries, they get caught. They get jumbled up with each other. Red blood cells are smooth. They slide by each other. They fold and squeeze through and nice and slippery. Sickle cell, all nasty, and they bind to each other, block off this so no oxygen gets to distal tissues. So what happens if that happens? Not only do you not get oxygen to distal tissues, but those red blood cells that are trapped there, they will lyse. They'll break open. Okay? And that can cause a whole host of other problems as well. So that's where we come into big differences in the globin chains when we have these types of mutations. All right, we know this. This is just a repeat. Oxygen binding to hemoglobin when you're in regions of high oxygen concentration, which is where? Lungs. You hold on to O2. You're sucking it up, right? But then when you get into areas of low oxygen, you release, right? And where is that? Tissue level, right? In the lungs, we call that what kind of respiration? External. At the tissue level? Internal. Good. All right. Iron is obviously very important. If you don't have iron, you're not going to carry oxygen, period. All right. So it's important in hemoglobin. It's also important for some other factors in your body, such as myoglobin. If you don't know what myoglobin is, it's like hemoglobin, but it's in muscle tissue. So it acts as an oxygen reserve. But iron's really important. So that means we store iron in our body, and maintaining those reserves of iron is going to be very critical in order to be able to continuously make red blood cells. Total iron in our body is about 4 to 5 grams. 65% of that is already being used in hemoglobin. So most of it's being used in hemoglobin. There's some being used for other things, but most of the rest is stored. Right? So most of it's being used, 2/3 of it's being used, 1/3 of it's being stored. All right, and it's stored as inside a protein called ferritin. And ferritin is in a couple different places, but most prominently in our reticuloendothelial system, RES. That's what RES is. Basically, that's a phagocyte system, which tells you that phagocytes eat the senescent red blood cells and then store the iron for them to be released later. All right, and we'll talk about where you find those and what they do later on. All right, so I'm looking at the slide and I don't like it. Yeah, no, don't worry about that slide. We're going to talk about all that stuff that's on the slide. It's just looking at that slide is confusing. So, where's iron absorbed from? So it comes, or where do we get iron? We get iron from our diet, right? And when we get iron from our diet, it is absorbed in the small intestine, all right, and attached to a protein called transferrin, all right. What's the symbol for iron? Fe. So when we see this term ferrin, we're talking about iron, right? So transferrin would be transporting iron. All right, so transferrin transports iron. And as it's being transported in the blood, it can be released to any cell, right? You can release iron to any cell. It's freely to be released as it's absorbed. Red blood cell precursors, that differentiation pathway that we talked about, have transferrin receptors on them. So what will happen is, as I'm making this way too complicated. That sucks. Let's say the orange is iron, and the blue is transferrin. As transferrin is carrying it, the iron, it will bind to the receptor on those red blood cells that are going through the pathway, and then it will endocytose the transferrin and the iron, and you'll use that iron to make hemoglobin within those red blood cells. All right, so I don't know. I think that's a neat mechanism. I come from a molecular biology background, though.
Just to be clear, when the iron binds to the apoptransferrin, that structure then becomes transferrin. Is that what that's saying?
Yes.
Okay.
Yeah, good point. All right. What Nick said, if you didn't hear it, when the protein is circulating around the blood without iron, it's called apotransferrin. When iron's bound, it's called transferrin. Okay, so that's what that little thing means. Once iron is bound, it's transferrin. Thank you for calling me out. Okay. one of the, aside from red blood cells, okay, so we know red blood cells need the iron, and that's where most of it's going to go. But iron will also go especially to hepatocytes. And where are hepatocytes located? Liver, good. To hepatocytes, and also those reticuloendothelial cells. When it does, when the iron makes it to, and again, it's being transferred by transferrin. When it makes it to hepatocytes, for example, the transferrin will transfer iron to apoferritin, and then when iron is attached to or combined with apoferritin, it becomes ferritin. All right? So back up a second. Any protein that deals with iron but doesn't have iron has apo in front of it. Once it has iron, you lose the apo. All right? And this is what ferritin looks like. So it's this big complex. These are all polypeptide chains right here. So it's a pretty big protein. And iron is kind of sequestered in the middle of that. All right, so what that effectively, and it's stored inside the hepatocytes. So keep it inside the hepatocytes. And you want to keep it in a structure like this so you don't have free iron messing up other things in the cell. All right, so it kind of holds it in this little ferritin sac, if you will. And the good thing about that ferritin sac is that ferritin can easily release it when it's needed. All right, so this is a good storage form. You can also find iron in a structure called hemosiderin. Hemo, blood. I don't remember what siderin stands for. That is also inside of cells, but it's a cluster of degraded ferritin plus iron plus some degraded heme, and it's insoluble and not easy to get the iron out of there. So although it is an iron storage form, if you have too much hemosiderin, that can cause problems. All right, so if you want iron stored, you want it stored as ferritin. You want as little hemosiderin as possible, because it can be problematic. That's more patho. All right, iron exchange, of course, like anything in your body, when it's low in the plasma, iron is released from ferritin. Put onto apoferritin, and then apoferritin becomes ferritin. Transferrin. Apotransferrin becomes transferrin, and then it's transported in the blood. All right, so released from ferritin, transferred to transferrin. Then it goes in the blood. All right, goes to red bone marrow, binds to those transferrin receptors on the red blood cell differentiation pathway, takes it in, we talked about this, gives it to the mitochondria, and incorporates it into heme. Okay, so now we just built this picture. If we have a deficiency of transferrin, what does transferrin do with iron? Transports it. Okay, if we have a deficiency of this, we can get something called hypochromic anemia. If we can't transport iron to where it needs to go to make hemoglobin, we get hypochromic anemia. And this is what that looks like. This would be a normal red blood cell. This would be a hypochromic red blood cell. Again, low color, right? Because you don't have that iron. Iron is what gives it, and oxygen bound gives it its color. So we know red blood cells have a 120-day lifespan. When they get towards the end of that 120-day lifespan, they are what we refer to as senescent. They're old. All right, and your liver and your spleen are able to detect that red blood cells are old, and when they do, the macrophages within them will, what do macrophages do? Macrophage, big eater, they'll eat the red blood cells and recycle everything, including storing the iron as ferritin.
Are the old ones, do the old RBCs, like, are they degrading as well? Is that why it's recognizable? Or is it just like,
Say that again.
if you said the senescent ones are old and it recognizes that, are like they starting to fall apart, or it just, there's something that says, okay, that's 120 days old roughly.
No, it's they. If I remember correctly, I think they just start to display things on their surface
Okay.
that will flag them for destruction, and macrophages will be like, "All right, I'm hungry." All right. So we take iron in through our food, and that can vary a lot throughout the day. But every day we actually lose a small amount of iron, about 0.6 grams. And how much iron do we have in our body total on a regular basis? What was that number we gave? Do you remember? Nope. 4 to 5 grams. So we lose about 0.6 grams, where was that, per day in men, and about 1.3 milligrams, not grams. 1.3 milligrams per day in women. Most of that loss is through the GI system, and of course women lose more through menses, because when you lose red blood cells, you lose iron. Okay? When we take it in, iron is absorbed through the small intestine. So we have this duodenum here, and we have our gallbladder here. This is not like even close to accurate to where these things should be located, but I want to get the point across. All right. And what does the gallbladder store? Bile. One of the components of bile is apotransferrin, right? And what does transferrin do? It transports, right? So what will happen is you'll squirt bile into the duodenum of the small intestine. Iron coming in will bind to the apotransferrin. That will go down to your small intestine, and then somewhere in your small intestine, the cells will have a receptor for transferrin. So transferrin will bind to that receptor. The enteric cell will endocytose it, and then we'll transport it into the blood, and then that's how you get iron back into your blood. All right? Again, another really important function of bile and the gallbladder. Does that make sense? Okay. So that's what all this says. I just like to draw pictures. So maximal absorption, only a couple milligrams per day. If you're only losing 0.6 per day, then absorbing only a couple milligrams is fine. And you'll store it, and get rid of any extra. All right. Let's finish red blood cells, then we'll take a break. Red blood cell destruction, when they get damaged, when they get worn out, they will get removed from the body. So they will detect through probably cell surface receptors. The macrophages will say, hey, you need to be destroyed. They'll phagocytize them, break them down. The previous slide says everything I'm going to say here. Here's your phagocyte. It'll take that red blood cell. It'll break it down into all the red blood cell parts. But importantly, the hemoglobin, you'll have the globin part, which is made up of protein, amino acids. It'll break that down, recycle the amino acids. It'll take the heme and make bilirubin out of it. All right? And it'll take the iron and store it as ferritin. All right? What happens to that bilirubin is it goes out, it gets out into the blood. attaches to albumin, makes it to the liver, it's conjugated with glucuronic acid to make it water soluble, and then it goes into bile. Okay? So effectively, we've recycled all the amino acids from globin, we've converted heme into something that we can get rid of, and we've stored the iron for later use. Really efficient system. So we make no red blood cells. And it's that bilirubin in the bile that gives poop its color, that gives urine its color. All right, but in order to do that, it has to be conjugated. So if you have liver problems and you can't get rid of bile effectively, does your poop have color? No. It has like a gray, ashy appearance almost. And what happens to that bilirubin? Builds up in your body and you get what? Jaundice. Good. All right, let's take a break. Because I need one too. All right, we're going to do the white blood cells, but I'm going to go through them really quickly because I want to spend more time on blood typing than I do white blood cells. White blood cells, they're not difficult. So, white blood cells, their main function is to protect against foreign invaders. And we have two general flavors, the granulocytes and agranulocytes. Granulocytes have granules. Anything that has granules is geared towards secreting. All right? So they have granules because they secrete things. Agranulocytes don't have granules, therefore they don't secrete. All right? Our most abundant, I don't care that you know the percentages of these, but know the relative abundance in relation to each other. The most abundant are neutrophils, also called PMNs or SEGs. You might see them like that different places. They are the most active. They are the first to arrive at the site of an infection. They are going to be doing the most damage to the foreign invaders, and they are going to be the dead bodies that are left over forming the pus afterwards. All right? They protect against all kinds of foreign invaders, including bacteria, fungi, viruses. The other type are much less abundant. You have two types, eosinophils and basophils. Eosinophils, I'm not even going to get into the morphology of them, but they defend against parasitic worms and also help with the moderation of allergic reactions. Do you know why they're called eosinophils? Because they're stained with eosin, which is a dye. Do you know why they're called basophils? Because they're stained with a basic dye. All right, basophils, they have a very similar function to mast cells in tissue. And mast cells in tissue are those tissues that, when irritated, release, help with inflammation by releasing histamine. Histamine makes things leaky, and heparin to inhibit blood clotting so that you have that leakiness perpetuated. All right? So this happens in the blood as opposed to mast cells that happen in the tissues. Now, there used to be a thought that mast cells were basophils that got out of the blood and went to tissues, but that's been proven not to be true. All right, agranulocytes, the biggest one, not the most numerous, is a monocyte. And monocytes are circulating around. When they get the signal, they will leave circulation, and as soon as they leave the circulation, they become macrophages. And they're kind of the scavengers that go looking for things to devour. And the other type are lymphocytes. And we'll get into this when we get into the lymphatics and immunity chapter. Those are our T cells and our B cells. Right? B cells are the ones that eventually will produce antibodies. T cells are the ones that will kind of regulate the immune system. And also one type that just kills everything. All right. The clinical importance of these is we can track them, and they can tell them things that they can tell us things that are going on inside the body. So we do what we call white blood cell counts, and there are two types. We have a total white blood cell count and a differential white blood cell count. So a total white blood cell count is the total amount of all white blood cells that you have. A differential would be how many of each kind do you have, right? Because if you have an increase in neutrophils, if you have an increase in eosinophils, that could tell you maybe we have a parasitic worm infection, right? So they can give you information about that. If you see an increase in white blood cells, total white blood cells, what could that mean? Infection. You might have an infection. You might have inflammation going on somewhere. Okay? So, I included what this might look like in a chart. So you will always have the counts of things, the results, the units they're in, and then, like I said, your reference range, like what's normal. And then, not only that, but they'll show you, they won't even make you read the reference range. They'll say, all right, this is high, or this is low. Okay, so the work is kind of done for you. Does that mean that you get a free pass and not have to know what that means if it's high or low? No, you still have to know that, but it takes some of the guesswork out. All right, platelets are also known as thrombocytes. Thrombo means clot, so clotting cells. They're not actually cells. What happens is that in your red bone marrow, you make these big cells called megakaryocytes. Megakaryocytes. The megakaryocytes will go into interstitial tissue attached to the outside of where capillaries are, kind of put little projections between the capillaries, and as blood is flowing past, they'll release parts of themselves into the blood. So literally, like that scene in Avengers Endgame. No, not Endgame. The Infinity War. Where everybody went, kind of turned to dust. The megakaryocytes, the platelets, will kind of pinch off and get into circulation that way. So eventually, this megakaryocyte will get smaller and smaller and smaller, die, and it'll be replaced. Okay, the job of platelets to basically stop blood flowing, hemostasis, so that we can repair blood vessels. We call that a platelet plug. We'll get into that later on. All right. As far as the development of white blood cells, same thing as red blood cells. You have the hematopoietic stem cell. When you get a certain growth factor binding to a receptor on a hematopoietic stem cell, it'll go down any one of those differentiation pathways depending on what's needed. So they develop in the bone marrow. They're going to stay there until they're actually needed. So it's not like red blood cells where you just constantly produce them. You produce them and they stay there until the need arises for those particular white blood cells. So the number of white blood cells in the bone marrow is usually 3 times what's in the blood. All right, and with that that large amount of storage of these white blood cells, you have enough for about a 6-day supply to replenish what was lost or what was used. So that's granulocytes and monocytes. Lymphocytes, on the other hand, they develop in your lymph organs, like your spleen and your lymph nodes. God, words are not coming to me. Lymph nodes, things like that. These other things like tonsils, pyre patches, those are just lymphoid tissue that will contain lymphocytes. And then megakaryocytes, of course, we talked about where they start and where they end up, on outsides of capillaries. Lifespan of white blood cells, the granulocytes in general, when they're circulating, about 4 to 8 hours. When they're in the tissues, about 4 to 5 days. Okay, so they live longer in the tissues than they do in the blood. All right. If you have infections or inflammation, if they're being used for what their purpose is, then you have shorter timelines because you're going to need more of them. All right. Macrophages, they circulate for about 10 to 20 hours. When they get into the tissues to do their thing, they can last for a long period of time because that's where they are doing their actual work, right? Chomping. They're Pac-Man. Lymphocytes, all the time. Okay, constantly being used and recirculated. They can last for your lifetime, right? Because one of the things, if you remember, and we'll talk about this when we get there, you have memory cells, memory T cells, memory B cells. Those stay and don't do anything until they're active again, right? And that's how you maintain immune competency for a lifetime. All right, then platelets are replaced every 10 days. So every 10 days, they wither down to nothing and get replaced. All right, blood typing. Who has had problems with this in the past? You? Okay. Okay. Hopefully we can help it make sense. I'm not going to cover it in like the detail that I would usually for undergrads because we don't have the time for that. But if you guys have questions about this and don't understand it, please feel free to come talk to me. I also, on Canvas, put all the statistics of the different blood types, what their prevalence is in the population, distribution by genetic background, and compatibility. So if you have trouble if you have trouble figuring out what's compatible with what, if you are this type, you can receive this type. Anytime you see a blood drop, that's what you can receive. All right? So let's get into what it is. All right, all red blood cells have antigens on their surface. Let's draw a red blood cell. And on the surface, we will have sugars. All right, on the surface, you will have these sugars. They're either glycolipids or glycoproteins. So it's the sugars that are important. It's the sugars that the immune system recognizes. It's the sugars that give you your blood type. All right? Do genetics code for sugars? Do genes code for sugars? No. Genes code for proteins. Okay, genes code for proteins. So if I have type A blood, and that means I have a certain type of sugar, what's going on? All right, what's going on is the type A gene codes for a protein that puts the sugar on the red blood cell. All right. The type A protein puts a type A sugar. We're not going to get into the different types and what the different branches of sugars are, but the type A protein will put the type A sugar on the surface. All right. That's ABO antigens for dummies. Not saying you're dummies, but that's like the condensed like Cliff Notes version. All right, so does that make sense? So that's how you have a type A genetic profile leading to a type A sugar. All right. The thing that people don't realize, though, is that these sugars are present on every cell of our body, not just red blood cells. But it's important to red blood cells because we do blood transfusions. All right. Those surface antigens, an old name for them are agglutinogens. Agglutinogen means antigen. So I will never use agglutinogen, but if you hear that word, it just means antigen. Because a lot of old farts out there still use it. All right, so we have different genes, A, B, O gene, and then we can have different genotypes. All right, O, O, A, O, and all that. We're not going to get into the genetics of this. We're more concerned with how the typing occurs. The agglutinins, which is another old term, are the immunoglobulins. And what's another name for immunoglobulins? Antibodies. Immunoglobulins are antibodies like IgA, IgG, IgE, things like that. All right? Our antibodies are anti-A and anti-B. So here we go. Our antigens on the surface are A and B. If we don't have the antigens on the surface, we have type O. Our antibodies are anti-A and anti-B. So what does that mean? If I have anti-A antibodies, what's that going to attack? Type A blood. If I have anti-B antibodies, what's that going to attack? Type B blood. Okay? So that gets us started. All right. The antibodies that we're talking about, mostly those are IgM and IgG, as opposed to the other types. That's what's going to be doing. These antibodies start developing about age 2 to 8 months. So you don't have them when you're born. They start developing when you're still just a little one, and they peak about 8 to 10 years. All right, and they respond to A and B antigens. Why do we have antibodies against A and B when we've never been exposed to A and B blood? Because turns out bacteria carry these same sugars. And we're eating things with bacteria all the time, right? So we develop these antibodies against these sugars because of the bacteria that we're taking in our bodies. All right? That's why we have anti-A and anti-B. That's where it comes from. Okay? Remember that, because when we start talking about Rh factor, it doesn't work that way. All right? So assume that everybody has anti-A or anti-B, or neither. We'll talk about why that is in a second. All right. So here is the summary of all this. If you are type A blood, you have A antigen and anti-B antibodies. Right? Why don't you have anti-A antibodies? Because you'd attack yourself. Doesn't make sense. If you are type B blood, you have B antigens on the surface of your red blood cells, and you have anti-A antibodies. And you don't have anti-B because you don't want to attack your own red blood cells. If your blood type is type AB, anybody in here AB? One. Lone wolf. Anybody in here type A? B? O? I think we've got you guys are lifesavers. You're a no?
No, I said thanks for the blood.
Oh, okay. All right. If you're type AB, you have both A and B antigens on the surface of your red blood cells. And if you have A and B antigens both on the surface of your red blood cells, you don't want antibodies. So you are not going to have any antibodies. Which means that you can receive blood from anybody, right? Because you don't have antibodies to attack that blood. If you have type O blood, you don't have any antigens on the surface. That's not 100% true because O is an antigen. It just is not immunogenic. So you don't have any antigens. None. Okay? But since you don't have any antigens, you will have both anti-A and anti-B antibodies. Okay? All this ring a bell? Yep. Okay, good. All right. So, why is this important? And why do we have blood type matching? Because if we give somebody type A blood, but they have type B blood, right? This would be the donor. This would be the recipient. Recipient. Jesus, that was difficult. Then the antibodies from this B will go and attack all those red blood cells. And the first thing that'll happen is it'll clump all them together. All right, because remember, I said this earlier. That's what antibodies do. They bind. So they're going to clump all those cells together. All right, and then what happens? All those red blood cells get destroyed. All right, and they travel in a complex, so not only do they get destroyed, but if they get to smaller blood vessels, what's going to happen? It's going to block those smaller blood vessels. Did you say clot? That is not a clot. That's called agglutination. All right, big difference. And I want to make sure you understand that terminology. Agglutination is the red blood cell antibody complex. A clot is something way different. Okay. All right, so this is what that looks like. We give blood to an incompatible person. Antibodies, antigens, they all clump together. This is what a normal blood smear looks like. This is what an agglutinated blood smear looks like. You have that going on in your blood, that just doesn't look good, right? So, lyse all the blood cells, cause problems with vascularity. All right, so we have to make sure that we match appropriately. If you have type A blood, you get type A blood. Okay? We talked about all of this. If you have type B blood, you get type B blood. If you have type O blood, who can you get blood from? Only O. If you have type AB blood, who can you get blood from? Anybody. Why? Because you don't have antibodies to attack red blood. Right? So universal donors would be O, because they can give to anybody. Universal recipients are AB. So, ideally, You want half your population to be O and half your population to be AB. So you can give blood to each other. It doesn't work that way. All right. Again, if you want the frequencies, how frequent is AB, O, AB negative? Like I have that chart in Canvas for you. All right, it's already there. I'm not going to hold you responsible for it, but it's always a question, so that's why it's there. All right, the other one. So we've talked about if you're AB, but you can also be AB positive or AB negative, right? So the negative, the positive or negative, it comes from the RH factor, the rhesus factor. It comes from rhesus monkeys. And that is, there are 6 antigens that are the RH factors. Actually, 3, C, D, and E. All right. The most common one is D. So we talk about the D antigen. All right. And the way this is inherited is you either have the big D gene or the little d gene, right? You have the big D gene, the antigen's expressed at high levels, and you have it on the surface of your red blood cells, and you're Rh positive. If you have a big D and a small d, since big D is dominant, this will also make you Rh positive. But if you have both recessive alleles, then what are you? Rh negative. That means you don't have the D antigen on the surface of your red blood cells. Okay? Who here is positive? Who's negative? Okay? All right. Under normal conditions, not being exposed to anybody else's blood, we don't have antibodies against the D antigen. We don't have antibodies against the D antigen. Do we have antibodies against A and B? Yes. Do we have antibodies against D? No. Why am I making a big deal out of this? Because the only way you're going to have antibodies against the D antigen is if you've been exposed to Rh positive blood. The only way you're going to have anti-D antibodies is if you've been exposed to Rh positive blood. That's only half true, though, right? Because if you're Rh positive, are you ever going to form anti-Rh antibodies? No, because you're going to attack your own red blood cells. Okay? So if you are Rh negative and you get exposed to Rh positive blood, that's the only time you're going to have anti-D antibodies, anti-Rh antibodies. That makes you sensitized. All right? Why is this important? Not why is this important? When are you going to be exposed to blood? When can you possibly be exposed to Rh positive blood? What are some conditions? Pardon? If your fetus is Rh positive and they are born, right? It's not going to happen in utero. It only happens when blood is mixed when you're born. When else might you be exposed to Rh positive blood? Blood transfusion. When else? Needle sticks, right? Drug users, things like that. Okay. That's the only way you're going to be exposed or have anti-Rh antibodies, anti-D antibodies. Okay? You're sensitized. All right. So, that happens the first time. That's a primary immune response. It's pretty weak. You're not going to have much of a reaction going on because it's the response isn't that big. But if you're ever exposed again, then you have a secondary immune response. All right, go back to your physiology from undergrad or wherever you took it. Secondary immune response, they are much stronger, much more antibodies, much more vigorous, much more dangerous if we're talking about D antigens. Okay? So, we only want to get exposed once. If we get exposed again, it's going to be big problems. Then you're going to have a big transfusion reaction. All right. So these are saying the same thing I just said. I'm not going to go through that. And the end product. If you're exposed that second time, you will agglutinate into the same thing that happens with A and B. All right. So when you're blood typing, you have to match A, you have to match B, you have to match RH. All right. Make sense? That's weird. All right, if I have A positive blood, who can I get blood from? I can get blood from all of them, right? I don't have anti-A antibodies. I'm not going to have anti-D antibodies. Right? So I can get blood from all of these. Can we back that up? If I am, what did I say, A positive? A positive, who can I get blood from? O positive, O negative, A positive, A negative. So this chart shows exactly what we did. Okay? If I'm O negative, who can I receive blood from? That's it, right? So if I'm O negative and I need blood, good luck. Okay? If I am AB negative, who can I receive blood from? Good. Negative all. Right? Universal recipient. But I can't receive positive blood because then I'll be exposed to the D antigen. And then I will never be able to accept that blood again. So you can do it one time. It won't be that big of a deal. But after that, so you may as well not ever be exposed. I don't know the regulations behind that. I mean, theoretically they could, but do they? I don't know.
Is there a way to know, like, before they give it to you, if you've been exposed, it would have a massive reaction, or is it just kind of like...
They'd have to test for it. Yep, they wouldn't know. I mean, they could say, you could say, I've had a baby, but you don't have no way of knowing. Like, you just have to test. Okay. So, on a test situation, if I give you a situation where I said, this is your blood type, which of these 5 choices can you receive blood from? You should be able to do that. Okay, that's the end game of this. All right, and you have that chart to help you. That will help you study. So what I would recommend is do some problems. Like write out, just like I did, donor recipient. If I have this type, who can I receive from? Then go to that chart and say, was I correct? Okay. All right, last thing, and you guys will actually get a similar lecture. Well, I don't know how they're going to do it since our instructor changed. Similar lecture to this again in your Clin Med class, I think in the spring. So, hemostasis. All right, that is not homeostasis. Hemostasis, blood stop. All right, blood stoppage. All right, so we're stop bleeding. Why do we want to stop bleeding? Why would our body want to stop bleeding? To, obviously, prevent the loss of blood. But why else would we want to stop bleeding when we have an injury?
Losing oxygen.
Pardon?
Infection?
No?
Maintain oxygen.
Pardon?
Maintain oxygen.
No? Pardon?
Your blood is clotted.
No? What's causing us to bleed? Injury to what? Blood vessel. Can we repair a blood vessel if blood is coursing through it? No. So we're going to stop bleeding to repair the blood vessel too. And the tissue. So, different mechanisms to stop bleeding, and they all work together. Vasoconstriction, what we call a blood vessel spasm, and it's not like, it's like it clamps down. All right. If you clamp down a hose, it's not going to pour out water. Platelet plug formation. So all these platelets we talked about, they plug up the vessel and prevent blood flow, which will allow the third step, blood coagulation, to happen. All right, so you most of the time need a platelet plug in order for coagulation to happen. And then you heal the vascular damage and you recanalize. What does that mean? Blood vessel is a canal, so you make it whole again. You make it able to course blood through. All right. So some key events. We have a severed vessel. We have that platelet plug appearing. Oh, nope, I'm sorry. Platelet plug appearing. Clotting factors are going to start to adhere to that platelet plug. You stop the blood flow. And then that clot retracts and blood gets going again. What's missing from here? Which step? The constriction, right? That's not there. So you would also have vasoconstriction here to minimize blood loss. Because really, if you're going to plug up a vessel, is it easier to plug up a vessel that's like shooting out or trickling out? Right? So you want to clamp that down. All right, so a blood vessel spasm is an intrinsic response to blood vessel damage. And but what I mean by intrinsic response is when you cut that smooth muscle, the smooth muscle clamps up. All right, you guys, never mind. So that's what's going to happen. Blood vessel is going to constrict to prevent blood loss and to allow platelet plugs to form. It can also be triggered by a central nervous system response that says, "Hey, there was damage here, and there's a reflex to cut off that, constrict that blood vessel." All right. Bottom line, you constrict, slows down blood loss. All right. Platelets. We've already talked about what they are, so I'm not going to go over this slide. Just to remind you that that's what's going to happen. Platelets form a platelet plug. So some characteristics about platelets to understand before we talk about the platelet plug is they have actin and myosin in them. Where else do you find actin and myosin? Muscle. And what does it cause muscle to do? Contract. Okay? So that means platelets can also contract. Okay? Platelets can also contract. They also have a contractile protein called thrombostenin. All right, and that acts with myosin and actin to contract. All right, they have residual endoplasmic reticulum and Golgi. All right, so what that means is that they can, ER and Golgi are geared towards secretion. All right, so they can secrete enzymes, they can secrete factors, they can, like growth factors, things like that. Certain ER can store calcium, like the sarcoplasmic reticulum in muscle. So they can also store calcium. Because calcium, if you don't have calcium in your body, in fact, if you have hypocalcemia, in some cases, if it's really, 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 really low, you can't clot effectively. All right? It also has mitochondria and enzymes in them, so they can produce ATP, because you need ATP to contract. All right? So platelets aren't just these, like we say that they're just these parts of cells that kind of just do nothing. They're very active when triggered. All right, so how does this happen? All right, here's our injury. All right, outside of blood vessels, we have extracellular matrix, and in that extracellular matrix is collagen. Right, that's the primary extracellular matrix protein. All right, so these cells that line the blood vessel are called endothelial cells. When they're injured, they're going to release a protein called von Willebrand factor. You guys have probably heard about this because there's something called von Willebrand disease. All right, that von Willebrand factor will bind to the collagen in the extracellular matrix. All right. Platelets have von Willebrand factor receptors. So what's going to happen is that the platelets are going to bind to the collagen, the von Willebrand factor attached to the collagen, and start to plug the blood vessel. Okay? It's going to occur on both sides. They're going to start to plug the blood vessel. When they do that, the platelets change. They change their shape. They swell, so they become bigger. Actually, there's your platelet. It will become bigger and spiky. Right? It'll take on a spiky appearance. All right? And when it takes on a spiky appearance, they can interact with each other and bond to each other through bridges with a protein called fibrinogen. All right? So when they do that, they become sticky, they stick to each other, they start to fill in. Right? And this platelet plug slowly works its way towards the center. All right? They also release platelet activating factor. So once a platelet binds to von Willebrand factor, binds to other platelets, it'll say, hey, platelets, come and play with me. And it'll attract more platelets to come and add to that plug. All right? And then you're going to stick more to each other. And effectively, you're making a platelet plug by doing that. All right? So that, in addition to the vasospasm, slows down blood so that you can make a clot. All right, so next is clot formation. So usually clot formation, it takes a long time to explain, but that platelet plug occurs within 10 to 15 seconds. It's pretty quick. Clot formation begins in about 15 to 20 seconds, especially with severe vascular trauma. So shortly after that platelet plug hasn't even formed, and the clot starts to happen. So it's a very quick process. You can have a clot that completely occludes this blood vessel in 3 to 6 minutes, but that's going to depend on the size of the damage, right, and the size of the vessel that's damaged. All right, so the clot happens pretty quick. In about a third of an hour to an hour, the clot will start to retract, pull things together, right, and including pulling ends of blood vessels together so they can be repaired. All right, then over 1 to 2 weeks, fibroblasts start to come in. They're going to lay down the matrix to repair that blood vessel, organize itself into fibrous tissue, and then the blood vessel can be repaired. Okay, so the point is the clot happens in minutes to an hour. The repair process takes days to weeks. All right, so what are the steps in blood clotting? This is where you have all those, diagrams, and you have factor 8 interacting with factor whatever, right? Which you guys love, right? You enjoy that. I know. Okay, key steps in blood clotting. We're going to see this again. This is actually the end of the pathway. All right? Key things that happen. You get, do you remember when we talked about trypsinogen? And then we cut it and it's active to trypsin. You convert prothrombin to thrombin, right? Similar concept. By prothrombin activator with calcium, that forms thrombin. Thrombin converts fibrinogen to fibrin, and with the help of calcium, that'll form fibrin fibers that attach to those platelets and kind of secure them down like a net. So you guys know when you go traveling and you got all that shit on the top of your car, then you strap it down with a net? That's what fibrin does. Straps everything down and makes sure that it stays in place so that this is completely occluded. Okay, so the question that usually gets asked, I don't like these pictures right here as much because they assume that the whole blood vessels cut, right? Is it always true that the whole blood vessels cut? No. Usually what happens is you have a situation like this where one part of it's cut, and that has to be repaired. So if you occlude the whole thing, blood doesn't get through, but if you only include part of it, blood's still going through, right? So you don't have to wait weeks to get a blood supply back. All right, so those are the key steps. Those are the end of the process. All right, they're going to form at the site, but then the clot can actually extend beyond the borders of where the platelet plug is to even further secure it. All right. The other thing that happens is thrombin, this protein that we went over before, can get trapped in the clot. All right. And as prothrombin is going by, it can convert more prothrombin into thrombin and like a positive feedback, right? So the more thrombin that gets trapped, the more prothrombin you're going to convert. Therefore, the more fibrinogen you're going to make and the bigger the clot is going to get. So one of the very few examples you have in biology, or at least physiology, of positive feedback mechanisms. The other one we discussed Tuesday with babies being born, right, and oxytocin. All right, so clot retraction, we talked about that, begins in about an hour. What happens with that is that you have all this fibrin bound to the platelets and the damaged blood vessel wall. Those platelets, remember, have actin-myosin. They can contract. So when they contract, they're going to pull all this together. So they're actually going to pull the broken blood vessel together as well, right? That's what we call clot retraction. And that happens by actin-myosin binding with each other and pulling them together. I wondered what clot retraction looks like, so I went to the internet and I found this. Like, this is blood. This is what a clot looks like when it retracts, right? I thought it was cool. All right. As the clot retracts, I hate the way they explain this. It says it expresses serum. Basically, you're squeezing all of the fluid out of the clot, and that is free to join the blood again. All right, so this is a summary of all the clotting pathways. There are two. We've already discussed this part right here, but you need to be familiar with the intrinsic versus the extrinsic clotting pathway. Did you guys go over this at some time before in your previous history? No? Okay, so again, this is one of these summary slides that you can come back to. And also, pay attention to places where anticoagulants get involved and vitamin K is involved. Okay, I'm not going to cover that in a whole lot of detail. This is a list of all your clotting factors. All right, because what their factor number is, the name is, because sometimes you have factor 8, factor 9, but those actually do have names to them too. We just refer to them as factor 8 or 9 because it's just easy to write. Okay, so I'm not going to go over these. I put them there for curiosity's sake, and when you're going through these, you can go back and say, oh, that's what that clotting factor is and what it's doing. Also, importantly, it shows the source. So remember where I said most of the clotting factors come from? Liver, all right? But there are some clotting factors that don't come from the liver, and those are listed here as well. All right, starting with the intrinsic clotting mechanism. What do I mean by intrinsic versus extrinsic? What does intrinsic mean? Inside, like it's intrinsic within the system. All the components are found in the blood. So you don't need anything else. Everything that's in the blood is already there. You don't need anything else from the outside. Usually this is triggered by trauma to the blood, like hemolysis. Remember we talked about blood cells when they're incompatible and they bind together and you have hemolysis. What that can do is trigger a clot, which can then block blood vessels. That's bad. Or exposure to collagen. Okay? Or exposure to collagen. So what happens is when blood comes in contact with a foreign surface, blood vessels open, collagen's out there. What's in the blood? Hagman factor. That's what Hageman, Hagman factor. Comes in contact, that's factor 7. Or not 7, 12. Comes in contact, it activates. It activates. Hagman factor activates. And then in the presence of calcium, that's going to activate prothrombin activator. And we already talked about that pathway. Prothrombin activator converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. That's that net that goes over the clot. All right, so that's what that looks like. 12, activated. Usually when they activate, we call them 12A. A stands for activated. Okay? That takes factor 11 and activates it. With calcium, that activates factor 9. And the rest of the pathway is what we've already talked about. Okay? What I did here is I did this pathway, but I included parts of that table highlighted that relate to what's in this pathway. Does that make sense? So that you could easily go back and you don't have to go back and forth between slides. Okay? Again, once thrombin is formed, that can, you don't need prothrombin activator. Thrombin can create more thrombin. Okay? Which will take it down the pathway and activate clotting cascades. All right? For the purposes of this class, that's what you need to know. When we get to Clin Med, we will have to know specifically what these factors are doing. Okay? So this is an introduction. All right, it's 10:03. Do you want? What else do you guys have today? When's that? 10:30. All right, just finish. Is that what I heard? Yeah. Muscle through. All right. Extrinsic clotting mechanism. All right. That happens by something outside of the blood vessels, outside of the blood. So you have trauma to the blood vessel and tissues outside of the blood vessel. Those tissues tell the blood, hey, we're damaged, stop blood flow. Okay? As opposed to everything in the blood being exposed and then automatically happening. The tissues are driving this. All right. So you have trauma. The tissue is driving this whole pathway. I'm going to go back real quick to this. So, more complicated. The intrinsic pathway is a little more complicated. There's more factors involved. The extrinsic pathway, a little less complicated, less factors involved, but they both converge right here with factor 10. All right, so factor 10 on represents what we call the common pathway. Okay, and that common pathway, again, prothrombin to thrombin, fibrinogen to fibrin. Those fibrin fibers cross-link everything. This past summer, no, it was December, actually, we went to Tarpon Springs, Florida. Anybody ever been there? It's a really neat place. It's Greek, and they harvest sponges there. And so I've never been so interested in sponges. I was like, how can you find a sponge interesting? But it was. But they had boats with just nets of sponges all over the place. And I took pictures, but the pictures didn't come out right. So I got this from the internet. What it reminded me of, this is what I think of when I'm on vacation. A fibrin mesh around the platelets. So the net around the sponges. Kind of cool. That's why that picture's there, in case you're like, what the fuck is that? All right. But it turns out that both pathways can work with each other. If you have tissue injury and the blood is exposed to the tissue, you're going to activate the extrinsic pathway, but the intrinsic pathway is activated too. So both can happen with each other, okay, at the same time. The extrinsic pathway can be very explosive. Like, what we mean by explosive is everything all happens at once, okay, within less than 15 seconds. The intrinsic pathway is a little bit slower. So extrinsic, about 15 seconds, boom, you got a clot. Intrinsic pathway, couple minutes. Okay? So definitely know the difference between those two. All right, vitamin K, important. We don't produce this vitamin by ourselves. It's usually produced by bacteria. It's fat-soluble vitamin, ADEK, right? And it is important for the production of certain clotting factors. I thought it was on this slide. Okay? Specifically, factor 7, 9, 10, and protein C, which we didn't cover, but I borrowed this from my other slide. So vitamin K is important for producing these factors. Remember, factor 10 is that where the common pathway starts. So if you don't have factor 10 because you have a vitamin K deficiency, you're going to have a hard time clotting. So this is why when babies are first born, they give them that vitamin K shot. Right? Have you guys heard of this before? They give them a vitamin K shot to make sure that they have the ability to produce clotting pathways efficiently. Especially babies that may come from malnourished backgrounds. They just treat all babies the same. All right. We don't want clots to stay forever. So we have to get rid of them. So we call it fibrinolysis. Lysine, fibrin. Breaking down fibrin. Okay? So this protein called plasminogen is trapped in the clot over a couple days. Something called tissue plasminogen activator, TPA, which is something that you can actually give to patients. They call it a clot buster. Will activate plasminogen to plasmin. Plasmin will start digesting fibrin, like little scissors that are cutting all those fibrin nets, freeing the sponges. Okay? And that will dissolve the clot, allow the platelets to either get eaten up by macrophages or go along circulation and eventually be degraded somewhere, and allow blood flow to be restored. This is a video showing what a clot looks like when it starts getting dissolved. Okay, so the more I think, if I remember with this correctly, the more opaque it becomes, the more the clot's being dissolved. I think that's what it's showing. I'll have to watch it again, and then I'll send you an announcement. Okay. The one thing that it produces is D-dimers. So when you're digesting fibrin, you produce these things called D-dimers. And that's an important diagnostic tool, because if you see D-dimers in a patient's profile, that means they have a clot somewhere that you are unaware of, and it's being, the normal process is to break it down, but it may not be being broke down effectively. Okay, so you look at those D-dimers to see if there's clotting occurring somewhere in the body. All right, when clots occur abnormally, we call it a thrombus. All right, so that's an abnormal clot formation. If that thrombus starts to move, we call it an embolus. Okay, so just terminology. All right, we have all these clotting factors in our blood. Why doesn't our blood clot all the time? Because we have protective factors. One, the endothelial surface of our blood vessels is shiny and smooth. You want that because you have a rough surface. That was what triggers clotting. That's what we learned, right? We also have a sugar coating on every cell. It's this thing called a glycocalyx. Nobody ever mentions this, ever. Have you guys heard of this before, glycocalyx? Yeah? Okay. That also provides like a slimy surface to the cells. Very smooth, easy for things to get across. Not a rough surface. And then we have factors in our blood that will block thrombin from becoming activated, because you don't want prothrombin to be activated. All right, you don't want prothrombin to be activated. All right, and then the final thing you guys are going to be doing as PAs is looking at blood coagulation tests. All right, so a couple of those tests. One is bleeding time. All right, you have a small cut. Stop bleeding in 1 to 6 minutes. If it doesn't stop bleeding in 1 to 6 minutes, that means you probably have a clotting problem. So what would you ask your patient? How long ago did this injury occur? And if it occurred hours ago and it's still weeping blood, all right, we better do some tests to make sure nothing else is going on, especially platelets for that. Clotting time. So we put blood in a tube. How long does it take to clot? Right, simple tube. Normally about 6 to 10 minutes. Not very effective test, right? If you're way out in podunk and you have nothing better to look at, you can do that. The big ones are prothrombin time. That's one of them. We call that PT. Okay. What you do is you have blood, and you add excess calcium and tissue factor to trigger the pathway. Which pathway, if you're adding tissue factor? Extrinsic pathway. Good. Or you just read it? No? PT time, usually that's about 12 seconds. So you're looking for a 12-second PT time. If it's longer than 12 seconds, what does that indicate? Clotting disorder. Okay? Associated with PT, you're going to see something called the INR. And an INR normally is the PT of your test, like what you did with your patient, over what a normal PT is. All right? And that ratio, if it's normal, should be between 0.9 and 1.3. All right? We use this to monitor, oh God, I want to get this right, anticoagulant therapy. We use it to monitor anticoagulant therapy. And if you see a INR above that, that means that anticoagulant is working fine, right? If it's lower than that, if it's normal, that means you need to add more anticoagulant. I want to say it's heparin.
Right.
Okay,
Yeah.
so it's heparin. That's why I use the generic term anticoagulant. We have tricks when we don't know things. I just didn't remember. The other one is partial thromboplastin, which you'll usually see as APTT, activated partial thromboplastin time. All right, that assesses both intrinsic and extrinsic pathways. Time is usually about 30 to 40 seconds. Why? Because it's a mixture of both intrinsic and extrinsic. But another diagnostic tool to look at clotting, especially when you're giving anticoagulant therapy. Okay? Heparin therapy. All right, so that's it. I think, right? That's the last slide? All right. Thank you for your patience. Thank you for being awesome students. And thank you for